Hello everyone, welcome to the biology video lecture. In this video lecture, we will discuss about an overview of the cell from the topic cell, the unit of life. Okay, so what is given in the textbook is that when you take a peel of an onion, okay, and when you take the peel and when you mount a slide of that peel and observe under the microscope, you will see that some rectangular structures you may see under the microscope, which are basically the cell, the epidermal cell. And if you stain this epidermal cell with a stain, you may see, let us consider the stain as acetocarmine, okay? It will stain the nucleus. So what you can see here is basically a cell which have no intercellular spaces. They are all compactly arranged, and then there is a nucleus, which is a structure present inside. So when we talk about nucleus and the cell which has a nucleus, those cells which have nucleus in them are called as a eukaryotic cell, whereas on the other hand, the cell which do not have nucleus in them, they are called as a prokaryotic cell. We can take the example of bacteria and the mycoplasmas, okay, in the prokaryotic cells, because they do not have a nucleus. So what is the main function of the nucleus is that the nucleus is a membrane bound structure, okay? It has a nuclear envelope, the nuclear membrane is present, inside which the genetic material, the DNA of that organism is compactly arranged, and with the help of the chromosomes. There is also one more structure which is called as the nucleolus, which is responsible for the synthesis of the rRNAs. Okay, so this is the nucleus and the nucleolus, and where the genetic material of the eukaryotic organism is placed. But that is not case in case of the prokaryotic cell. In the prokaryotic cells, the genetic material, that is the DNA, it is present. This is also compactly arranged, but this is arranged in the cytoplasm. There is no separate envelope for this as a nuclear envelope that is found in the eukaryotic cell, right? So that is the first difference when we talk about a prokaryotic and a eukaryotic cell. Along with that, when we talk about the eukaryotic cell outside the nucleus, okay, there is a cytoplasm. This cytoplasm is basically the cytoskeleton. It is made up of different sorts of proteins that we will study obviously in the upcoming lectures. So this gives a skeleton system, it gives a support to that cytoplasm, and in this cytoskeleton there are different types of organelles that are situated over here. The organelles we can see in the labels like mitochondria, okay, then the peroxisomes, the Golgi body, the lysosomes, okay, the endoplasmic reticulum, okay. So all these are present and they are limited only to the eukaryotic cell, but they are not present in the prokaryotic cell. Okay, what is common among them, common among both of these, are the ribosomes. Okay, ribosomes are basically very tiny structures that are around 10 to 20 nanometers in size, and these structures are present in the prokaryotic cell as well as in the eukaryotic cell, because they are used in the synthesis of proteins. That process where the proteins are synthesized is called as the translation process. Okay, so for translation ribosomes are required. When we talk about the prokaryotic cell, a bacterial cell, the ribosomes are dispersed in the cytoplasm. When we talk about the eukaryotic cell, yes, the ribosomes are dispersed in the cytoplasm, but in the eukaryotic cell, the organelles such as mitochondria and the organelles such as the chloroplast, they also have their own ribosomes. They also have their own ribosomes. Okay, now when we talk about mitochondria and chloroplast also have their own circular DNA molecules. They have their own mRNAs and they can synthesize their own proteins. Some proteins, not all proteins, right? We will see in the upcoming topics that how the mitochondria produces its own enzymes which are required for which are required in aerobic respiration for the production of ATP, the Krebs cycle. We will also see that how the chloroplast has to produce its own enzymes, that is the proteins, to carry out the dark reaction, to carry on the electron transport chain, and so on. Okay, so this is in case of mitochondria and chloroplast, and that's why, as they have their own ribosomes, they have their own DNA, they have their own proteins, these two organelles, they are called as semi autonomous organelles. Semi-autonomous means, when we talk about autonomous, automatic, on their own. Semi-autonomous, that is, some, for some proteins and for some enzymes, they are dependent on the cell. But for the other enzymes and the proteins, they can prepare their own. So this is in case of mitochondria and the chloroplast. But these are not present in the prokaryotic cell. It is absent in the prokaryotic cell. Okay, so these are some of the things that we can use to discriminate between a prokaryotic cell and a eukaryotic cell. Okay, so now let us talk about animal cell and a plant cell. There is a fine distinction that when we talk about the animal cells and the plant cells, animal cells have a structure that is called as centriole, which helps during the time of cell division. But plants, they do not have the centriole. Okay, plants have large vacuoles in them. Animals don't have vacuoles. Okay, whereas animals, or we can say, sorry, whereas the plant cells, they have different types of plastids like chloroplast, elioplast, aleuroplast. These animal cells, they do not have the plastids in them, right? So this is a small discrimination between a plant cell and an animal cell. Later on, when we unravel the topic, obviously we are going to study, and we are going to study more points that will be discriminating between the animal cells and the plant cell. Right, now let us talk about the shapes, the sizes of the cell that is there in the syllabus. When we talk about the smallest cells, okay, the smallest cells are considered to be the mycoplasmas, okay, and PPLO, that is a pluro pneumonia like organisms. Okay, so this PPLO or the mycoplasmas, they range around in between 0.03 micrometer. But when we talk about the bacterial cell, it is around 3 to 5 micrometer. Okay, when we talk about the largest cell, okay, the largest isolated single cell, it is the egg of the ostrich. Okay, that is the largest cell. When we talk about the RBCs, okay, RBCs are around 7 micrometer, sorry, 7.0 micrometer in diameter. So that is about the RBCs. Okay, now when we talk about the cells and their shape, okay, when we talk about the shapes of the cell, okay, the shape of the cell may be disc like, the shape of the cell may be disc like, okay, that is, we are talking about the shapes. The shape might be polygonal, the shape columnar, the shape can be cuboidal. Okay, so these are the different types of shapes that the cells may have depending upon the function that they carry on. Okay, so the shape of the cell as it is given in the textbook, it may vary with the function that they perform. So this is about an overview of the cell. In the next video lecture, we will talk about details of a prokaryotic cell, and then we will start studying about the eukaryotic cell and the organelles, plasma membrane, and the cell wall of the eukaryotic cell. So thank you so much for watching the video. Thank you.